Hey guys, this is Mr. Pfeiffer, and welcome to Honors Biology 1, the Flipped Classroom Edition. If you're joining me from Mr. Hamilton or Mrs. Wickman's class, welcome. I haven't had the pleasure of meeting everybody yet, but all three of us are going to use the same videos, and that's just to ensure that all students are receiving an equitable and consistent education while they're in this class. One thing I'd like to say before getting into the content is all of these lectures are the content delivery mode of this class, and so we're not necessarily going to be opening PowerPoints, and we're not necessarily going to be reviewing all content in class, and so it's going to be vital for you to watch all of these lectures in its entirety, take good notes, and come to class with questions. And then make sure you get answers to those questions, because I don't know about the other teachers, but I know that I can't read minds, and if one of my students has a question and they don't ask it, it usually goes unanswered. Okay? So watch lectures, take good notes, come to class with questions, stay caught up, stay current on not only the online lectures but the online quizzes, and stay caught up on your readings. Okay? This class is going to be a lot of information in a really short amount of time, but it's going to be fun. Okay, this flipped classroom model is going to open up a lot of class time, and it's going to ensure that we have more time for labs, for hands-on activities, for model building, for elaboration and extension activities, more review of content in class, more review before exams, more quizzes, which may not sound beneficial to you, but it definitely is for us. And I know I've told my kids, but I'd like to tell everybody now that last year was the first year we implemented the flipped classroom, and our overall grades increased on every exam across the board, and every increase was 5%. So all the students last year scored a whole half letter grade higher on every exam from start to finish, including the final exams. Okay, so if you really buy into this program, really buy into this model, I think it's really going to pay off for a lot of you. Okay, this lecture is going to be over sections 1 and 2 in chapter 1, and it's probably going to be about 15 minutes. It really just goes through the scientific method, kind of the way science works, the way science doesn't work, and we'll leave looking at 2 experiments that really pave the way for biology and our understanding of the world and our understanding of life by disproving spontaneous generation. Okay, so again, welcome, and let's get started. Everybody in their class should have received a Chapter 1 vocabulary and objective sheet, and the vocabulary and objective sheet is really important for you. It's the driving kind of pre-study guide, or the guide that really kind of drives your focus, or should allow you to focus more specifically on specific objectives. Okay, all of the objectives right in here are going to be important, and this lecture is really going to go through this one, this one, this one, and this one. These last 2 are going to be talked about in the second lecture of chapter 1. Okay, and so after this lecture you should be able to actually answer these top 4 objectives in quite a bit of detail. Okay, so that's something to kind of drive your focus as we go through this. Vocabulary, these are the vocabulary words that are going to be important for you to know, not only for the vocab quiz that'll be in about 2.5 weeks, but really for the test. Okay, all of these words are going to be used in class almost daily, and so if you don't understand the vocabulary and you don't understand the objectives, it's going to be really hard for you to really focus on what's important in this class. Okay, so what is the goal of science? Science is a discipline that helps explain the way the world works and helps explain the way the universe works. One of the things that makes science so great is that there are a lot of subtopics of science, or a lot of genres of science. You've got chemistry and geology and astronomy and biology, and then even within those you've got microbiology and botany and genetics and cell biology and ecology and those kinds of things. And so it's really a science, or it's a field that a lot of people have interest in, and it's a field that a lot of people specialize in, and that really helps to create this body of knowledge, and this body of knowledge is huge. I mean, we have really collected a lot of data over hundreds of years, and that data started, thousands of years ago with a lot of the topics that we now know. Okay, just as an example, a few hundred years ago they thought the world was flat. We now know that is incorrect. A couple hundred years ago they thought the Earth revolved around the sun, or they thought the sun revolved around the Earth, and we know that is not correct. Okay, and so science is a body of knowledge that helps explain the way the natural world works. One of the things that makes science great also is that it's refutable. Okay, there are people that feel a certain way, and there are people that feel the exact opposite way, but science has a way of ensuring data is collected properly and ensuring that data is analyzed and interpreted properly. And so it's really easy to understand science when you keep a skeptical or refutable mindset. And science is a process. Okay? So experimental design. Science is one of those things that is kind of step by step, and it's sequential, and the experimental design needs to be done sequentially in order for the experiment to be run properly, and in order for data to be collected and analyzed, and ultimately for a conclusion to be generated. The first thing you have to start with is an observation. Okay, and observations are those things that you use senses for. So, like sense. Senses like eyesight and taste and touch and hearing and smelling and those kinds of things are pivotal in your understanding of the world around you. Okay, so you make an observation that leads to a problem. Because of this observation, you now have a question. Sometimes you refer to the problem as a kind of a guiding question or experimental question, or a problem that needs to be addressed, needs to be discussed. Okay. Once you have a problem identified, it leads to a hypothesis, and those hypotheses need to be a cause and effect. A lot of times you're going to hear it discussed as an if-then because statement. If this happens, then it causes this because of that. Okay, and I know that's very general and generic, but if-then because statement, it shows cause and effect. That's hypothesis. Then you have to identify variables, and those variables can be constants, they can be controls, they can be independent variable, they can be dependent variable, and all of those help to create a cohesive, very controlled experiment. One term I like to use interchangeably with independent is the manipulated variable, because that's the variable that you physically change. The dependent variable is the one that responds to your change in the independent variable. Okay, so for instance, you're doing your pill bug lab, and some of you might have actually done this, but if I want to change the color of light, the independent variable would be the color of light, okay, because I physically manipulate the color of light. In one chamber I put green light, and in the other chamber I put red light, and in the third chamber I put purple light, and in the fourth chamber maybe I put orange light. Okay, I manipulate the light colors. That is the independent variable. The dependent variable is the variable that depends on the independent, and in the pill bug experiment, the dependent variable is pill bug choice or pill bug numbers. Okay, that is what you physically measured. I think everybody counted the number of pill bugs that ended up in each chamber, okay, and the number of pill bugs that ended up in each chamber was directly dependent on whatever it was that you manipulated, okay, whether that was color of light, intensity of light, soil salinity, soil pH, temperature. It could have been a whole host of things, and I was quite impressed with the amount of manipulated variables, the amount of independent variables that my classes and all the classes came up with. Once you have your identified or your variables identified, you're going to need to design the experiment, and this is when you actually write the procedures. And if you remember from the peanut butter and jelly experiment or the peanut butter and jelly demo, that you can't assume the reader knows what you're talking about. Okay, you cannot assume anything. You have to write very descriptive, very articulate set of procedures in order to adequately set up or or create an experiment. Okay. Once you set up the procedures, follow the procedures, you have to record data. This would be recording raw data that's typically in a data table. Once you record your data table, or record the data in the data table, then you need to start processing data. And that's when you can add your standard deviations, you can add your error bars, you can add your variance, you can add percent uncertainty. Okay, and so once you process the data, you can start analyzing that data. Okay, once you analyze the data, then you're able to draw conclusions. And because you draw conclusions based on an interpretation of the data, it's more scientifically accepted, or it's more accepted within the scientific community because, again, it's based on data. Okay? Good. So you are going to need to know these steps, and if you're in my class, I wrote out even more steps. Some of the steps that I wrote out are actually kind of combined. Like I had the independent-dependent variable, I had control, I had constants, I had within the analyzing data, I had processing data and those kinds of things. And so you can really have as much as like 13 steps, but you could probably even kind of combine these in order to go down to like 6 steps. But you need to know the actual process in a sequential step-by-step order. Okay, so now we have a problem. We make an observation that our flashlight is not working. Okay, we use our eyes, we physically turn on the switch and no light comes out. And because no light comes out, the flashlight is useless. Okay? We used our senses to make this observation. When you make an observation, it leads to a problem, and our problem would be we're stranded by ourselves, and we're in the dark, and your flashlight is not working. What is the potential problem and a correct hypothesis that corresponds to the issue? The problem is your flashlight's not working. Okay? The correct hypothesis could be a whole slew of things. Some of the hypotheses or problems could be maybe the batteries are dead. All right, that's probably the first thing that came into your mind. Second thing that came into your mind is probably the bulb is out or broken. Those are probably the top 2 reasons why a flashlight wouldn't be working. Maybe it's ghosts. Maybe wires from the batteries to the bulb went bad. Okay, maybe the batteries aren't dead. Maybe the bulb isn't broken, but maybe the wires from the batteries to the bulb went bad. Maybe there's a short in the wire. Maybe the insulation between the wires kind of rubbed together and rubbed off, okay, leading to a short. Maybe the batteries are corroded. Maybe the wires are good, but the battery just for some reason isn't contacting the terminals very good, okay? It could be ghosts. Never know. It could be that the switch went bad, okay? It could be a lot of things. It might be ghosts. Let's just assume that our 2 hypotheses that are probably most likely to be true are dead batteries and a burnout bulb. Okay, so we made an observation. The flashlight isn't working. We used our senses to do that. We question or create a problem. I wonder what it is that is causing this flashlight to be broken, or I wonder what it is that is causing this flashlight not to work. We come up with 2 different hypotheses. One would be dead batteries, one would be burnout bulb. And then we set up an experiment in order to test those 2 hypotheses. If it is dead batteries, we're going to predict that replacing the batteries will fix the problem. And so you go about testing that prediction. We put new batteries in the flashlight and test, and all of a sudden, no, it doesn't work. And so we are going to have to go back to the drawing board. We have to test our second hypothesis. Maybe it's a burnout bulb. Okay, so we replaced the bulb and it worked. Okay, so in this case, the test does not falsify our hypothesis, which means it accepts, or we accept the hypothesis. In this case, we falsify the hypothesis because remember, we thought it was dead batteries and it wasn't. So we reject this hypothesis and we accept this hypothesis. Okay? It must not have been ghosts this time. That shows the whole process. Remember, it's sequential, step by step. And so what happens when experiments are not possible? Okay, we have to use models. This is a model rendering of a specific tissue in plants. Okay, but what happens if we want to study viral infection rates and it's unethical to infect a population of people with a virus just to see how fast it spreads through a country. Okay, there are scientists that are studying viral infection rates. However, like I said, it's unethical to actually test that in a real-world setting, and so what you're going to want to do is you're going to want to set up a computer model with the infection rate to see how fast it would spread through that country. Again, that is kind of a fake way of finding it, but it's the most ethical in that case. Okay, and then experimentation leads me to this theory concept, right? How many people have heard in the hallway people say, my theory on why we don't get good food in the lunchroom is this? Okay, is that the real meaning of theory? Well, kind of, right? There's a big T theory and there's a small t theory. When theory is capitalized, that's big T theory, and that's a generally highly accepted hypothesis, okay? Like the theory of relativity, or cell theory, or the theory of evolution. Okay, these are things that aren't, fact, but they're so highly accepted that you can't really refute it at this point. Okay, are there things that will happen in the future that will lead us to a greater understanding of those things? Yes, but at this point we can't refute it, so it's generally accepted. When theories become even more accepted or basically proven right, it becomes a law, and laws would be those things like gravity, right? Law of gravity. Gravity is fact. It happens, it's here, it's everywhere. Okay? And so theory is really important. Big T theory is a highly accepted set of hypotheses that is accepted based on data. Little T theory is all about your kind of informal language in a hallway, and it's just kind of your hypothesis, not really a widely accepted, highly accepted, based on data, accepted scientific phenomenon. Okay, now, I told you at the beginning we would talk about this idea of spontaneous generation, and 500 years ago it was believed that organisms were created from non-living things. In fact, there were recipes on how to make organisms. There was a poet that wrote, like 500 years ago, the only way to make bees, or a recipe to make bees, was you build a shed, you hang an animal, like a bull, in it early spring, and by fall you would have bees. Now, was it the bull carcass that was causing the bees to form, or was the carcass just attracting the bees and they were building a hive in the shed? That's probably more realistic, right? And so this is an example of spontaneous generation. You put dirty shirts together in a pile, you sprinkle a bunch of wheat on the shirts, and 21 days later, all of a sudden you have mice. Okay, spontaneous generation says that mice and other organisms just spontaneously appear from non-living things. Okay, that is very false. What is actually happening in here? Dirty shirts and wheat are attracting the mice, and it gives them 21 days to find a food source in the wheat and a shelter in the dirty shirts. Okay, so what are the tests that kind of came about to disprove this spontaneous generation? Okay, well, Francisco Redi, designed an experiment to test why these flies and maggots were spontaneously appearing on old meat. Okay, because prior to the 1600s, people thought that meat that was left out unrefrigerated would just spontaneously erupt into these flies and maggots. Okay, so he set up an experiment. He had meat that he left out in open jars, and because they were left out in open jars, he found that the open jars produced flies and maggots. Now, he controlled his experiment. He then changed a variable, and in the next experimental trial, he put lids on the containers, and because they are not open to the air, and because the flies couldn't get to them to lay their eggs, which ultimately produced the flies, there was no flies that produced, the meat did not produce flies in the tightly sealed jars. When he then further experimented, he put mesh that was too small for the flies to get to, but still allowed the smell of the meat to escape. the flies were still attracted. However, they couldn't get to the meat, and so they laid their eggs and larvae on the mesh because they thought the mesh was the meat. Okay? How does this test in 1668 help disprove spontaneous generation? One, it proves that when flies are able to touch the meat, they lay their eggs and the flies are basically born. When flies cannot get to the meat does not produce flies or maggots. Okay? And Pasteur's experiment 200 years later, in 1864, was basically instrumental in producing our ultimate disproval of spontaneous generation. He boiled a bunch of broth, okay? That was to sterilize it, kill all the bacteria in it, and because he had this really long swan-necked flask, which creates an airlock and doesn't allow bacteria to get in, there was no growth of bacteria no matter how long he waited. When he took this control group or controlled experiment and tested it by doing the same amount of boiling but breaking off the lid, because bacteria now was able to enter it, he waited the same amount of time or even less, and microbial growth was the result. Okay? So this also disproves spontaneous generation, because when bacteria can enter broth, it grows. When it cannot, it doesn't grow. Okay? The broth was not producing bacteria. The bacteria were getting in and proliferating to the point where we now can see them. Okay? So that's all we're going to do here in the first lecture. Make sure you took good notes. Make sure you bring questions that may have come up as a result of some of the content in this lecture. Like I said, the content in this first one really wasn't that hard. It shouldn't be that difficult, but this is just kind of to get us into what is biology, get us into what science is, the goal of science, and then this idea of spontaneous generation and what was able, or what happened in the scientific community in order to disprove that. Okay? I'll see you next time. We'll talk about characteristics of living things. Have a good night.